All right, so we're getting into oxidative phosphorylation today. That's going to be that other side of the coin. If you can think of one side as being the dammed up water going through the channel, then the other side is the turn wheel that generates the power on the generator, okay? And a good analogy for this is actually something like this, which is a water wheel. And so we have potential energy being converted to, in this case, kinetic energy, right? And then you can use that kinetic energy for a variety of purposes, whether that is generating electricity or whether that is grinding down flour, whatever. But right here, you can actually see that overall process, though we'll have some better figures that will help out. Right here, again, the intramembrane space is where we're going to gather the H+. It's going to then use that potential energy of the H+ needing to get away from the other positive charges to go through into the matrix, which is going to be what turns this wheel right here, okay? It works exactly like a mechanism would in a car, water wheel, generator, any of those you want to think about, okay? Crankshaft, whatever works in your head. So take your favorite analogy in your head. Which one of those works? Which are you guys most familiar with? Water wheel, crankshaft? Huh? Water wheel? Okay. Or car, drivetrain. Could also be. Any of them will work. Respiratory chain. This is the electron transport chain for large protein complexes that are going to be embedded in the inner mitochondrial membrane. Oxidative phosphorylation is a set of electron transfer reactions. It's going to capture the energy of high energy electrons that are being transported by the NADH and FADH2. This takes place in the electron transport chain, which is this overall process right here. Ultimately generates ATP and reduces oxygen to water. The flow of electrons from reduced carriers such as NADH is highly exergonic. Remember that exergonic being the produces, gives free energy, right? Okay, and so this is that overall process, NADH plus 1/2 of an O2 plus H plus goes to water and NAD plus. Notice that has a negative 220.1 kilojoules per mole. What would happen if you tried to do this thermally instead of electrostatic potential? That's the sort of energy output that like burning gasoline has, right? Actually, it's a little larger. So if you did this thermally, you just night, right? Boom. You're gone. So the complexes of electron transport chain, these are going to be used to release the energy to pump protons out of the mitochondrial matrix into the inner membrane. This is going to generate a pH gradient and a transmembrane electron potential that will create a proton motive force that is used to power the synthesis of ATP. Note that this is actually very, very, very similar to how a lithium-ion battery actually operates, okay? Or any of the fuel cells also operate quite similar. Batteries operate quite similar to this, okay? So we're essentially just generating our own potential on one side. We're then using that potential a bit like a waterfall would, right? Okay. Now, the ADP plus a orthophosphate plus a H plus gives you ATP and water. This delta G naught prime is positive 30.5 kilojoules per mole. By the way, you'll notice that's just the reverse of the other way we did this. So, you know, if you go the opposite way, you just take the sign and flip it, right? There you go. In case you forgot. Now, note that this number is what compared to that number? A lot smaller, right? Okay? Which is why you don't just generate one ATP for every NADH you consume, right? Typically, you're going to generate about 2.5. Is that super efficient? No, it's actually pretty good, to be honest. Like, do you know what the efficiency of a typical engine is? Like in your car? Yeah, they're in the 20s to 30%. 30% would be really good. Okay. There's a hard cap in the 40% that you can't actually get to because the ambient temperature is too high. Yeah. Theoretically, if you design one to operate in a 0 Kelvin environment, or close to, you could get higher. But so this right here, oxidative phosphorylation, is going to be fundamentally the combination of two processes. We're going to have the pumping of the H pluses to build up an electromotive force. Okay? Or another way of saying that is to build up potential on one side, right? And then we're going to have that potential, which is going to drive our water wheel, we decided? Okay. Water wheel. Crankshaft, whatever the hell you want to do. And this is going to be what is actually literally going to press and cause them to bond the orthophosphates and ADPs to give you ATP. Okay? I am not joking. It is literally a press. You guys ever seen a press work? No? You've never seen like how it's made and they have that line of stuff and like they take the metal or whatever, if they're making sheet metal or whatever they're doing, and it's literally thunk. And then it does what it does. Yeah, that's what this is. Okay? It is literally phosphate, ADP, now you're bonding. Okay? Because you have to remember, when you force those things together, then they actually overcome the electrons coming together. And once they overcome that, if you're in the correct angle, the protons are going to start being attracted to the electrons, right? And so it is very literally thunk pressed. Okay? All right. And so, anybody know what this TCA right here is? That's another name for the citrus acid cycle, okay? So it's just the cycle we'd already been talking about right here. Okay? Notice it's the one that actually produces the CO2. The CO2 production and the oxygen consumption are actually separate processes, okay? They're subdivided. It's the source of your electrons that's going to be pulling the H pluses up here, okay? All right. So respiration, this is going to be the cellular respiration specifically, is going to be the generation of high transfer potential electrons via the citric acid cycle, their flow through the respiratory chain, and the accompanying synthesis of ATP. This is that entire process because this is going to involve the CO2 on the citric acid cycle portion. And then we go and transfer the electrons, and then we do the ATP synthesis, and that is eventually going to give us the oxygen consumption, right? Okay. So again, the citric acid cycle, the electron transport chain, and the ATP synthesis, these are going to occur in the mitochondria. Your mitochondria are going to have two membranes. You have an outer membrane, then you have an extensive, highly folded inner membrane. Why would this need to be highly folded, by the way? Let's do a little bit of critical thinking there. No, you got it. Like, if you're going, why is something the way it is in biology? More surface area. Why is something like it is in cell biologies? Why is it like it is in biology? More surface area. Yeah. Like, surface area can be the answer for probably about 75% of questions in biology, if you are vague enough about it. Yeah. So this is going to create an intermembrane space, right? It's a space between those two membranes. And again, that more surface area for the inner membrane means you have more spots that you can perform these processes on, right? You can pump protons through. The matrix is going to be the compartment that's bound by the inner membrane. And that's where that citrus acid cycle happened. And that's also where some of the different conversions happen. And we talked about all those things before, right? So the most citrus acid cycle and fatty acid oxidation reactions are going to take place in the mitochondrial membrane, or matrix, excuse me, not membrane. The oxidative phosphorylation is going to take place in the inner mitochondrial membrane. And again, this is what this looks like here. So again, here's the inner membrane, outer membrane's the blue part. The matrix is within the inner membrane. And then the inner membrane space is between the two membranes. Okay? And this right here would be the cristea, right there. Okay. So the outer mitochondrial membrane is going to be permeable to most small molecules and ions because of the presence of mitochondrial porin, which is the voltage-dependent anion channel. The ion inner membrane is going to be folded into internal ridges and tube-like structures. Those are the cristea. They increase the surface area to create more sites for the oxidative phosphorylation. They're impermeable to most ions and polar molecules. They're going to have transporter shuttle metabolites to shuttle metabolites across the membrane. It'll have two sides. It'll have the matrix side and the cytoplasmic side. Remember, the matrix in this case is quite different than what we would normally consider the inner part of a membrane. Okay? It's very different than our cytoplasmic cell cytoplasm. Okay? It's the mitochondria is quite a unique one. The mitochondria are going to be semi-autonomous organelles. These live in an endosymbiotic relationship with the host cell. They contain their own DNA. They probably were their own organism, but they are so integrated now that they're endosymbiotic. They're symbiotic to an incredible degree with us. And they actually dictate many things about what we think as our own lives. The endosymbiotic theory is the hypothesis that an endosymbiotic event occurred in the evolution of the eukaryotic organisms, in which the free-living organism capable of oxidative phosphorylation was actually engulfed by another cell, survived, and kept reproducing with that other cell, and is now tightly incorporated in almost every aspect. Okay? The sequence of data suggests that all mitochondria are descendants of an ancestor of Rickettsia, God, I'm terrible at Polish, Prowazeki, anyways, you can look it up. I'm sure that somebody actually pronounced it correctly at some point in life. Okay? Terrible at Polish names. The result of a single endosymbiotic event, they are highly conserved. They change very slowly over time. That's why you can trace DNA from a mother event back 10,000 years, no problem. Okay? The most bacterial-like mitochondrial genome is that of actually the protozoan Americana. The R. Americana, which would have been so much easier about myself, I don't know why I do this to myself. Genome encodes less than 2% of the protein encoding genes of E. coli. All mitochondrial genomes have approximately the same 2% of bacterial genes. This actually suggests that the endosymbiotic event occurred just once in evolution. So oxidative phosphorylation is going to be the electron transfer potential of NADH or FADH2. This is going to be converted into the phosphoryl transfer potential of our ATP. You can think of this as an energy transformation pathway. Okay? much like a generator would be taking the steam kinetic energy and transferring it to electronic energy, electrons, right? Or electricity, as well, most people like to say. So phosphoryl transfer potential, this is the delta G naught prime for the hydrolysis of the activated phosphoryl compound. The electron transfer potential is going to be the E naught prime, E naught zero. The reduction potential or redox potential or oxidation reduction potential, it's the same E naught, by the way, as you guys remember from Gen Chem 2, or if you remember that at all. Same one. Okay. This measures a molecule's tendency to donate or accept electrons, and you may remember it from your oxidation reduction work that you did in Gen Chem 2. I believe it showed up a little bit in organic, but only a very, very small amount. Redox couple, this is a substance that can exist in the oxidized form. Let's call that X, and a reduced form, that would be X minus. And so you typically would designate that as whatever it was before when it's neutral, and then 2 dots between it. Typically that denotes a barrier or salt bridge, by the way, some sort of membrane that can be exchanged across. And then this would be the X minus. So for example, we could have HNO3 and NO3 minus, right? Okay. The reduction potential can be determined by measuring the electromotive force generated by an apparatus. This is called a sample half cell. This is connected to a standard reference half cell. Is this sounding a little bit familiar? Okay. Remember that when you did your batteries and you did your half cells back in Gen Chem 2, they had a semi-permeable membrane that allowed the free exchange of anions across it, right? Guess what? We also have something similar in this case, don't we? So we have semi-permeable membrane that we can exchange across. And so that's actually what you're seeing here. You're seeing a decoupled half cells that only allow one part of the energy to transform across one section of it, right? In the normal case where we're looking at half cells in Gen Chem, that would have been the wire, right? And so we could generate electricity. In the case of these, it's going to be the ATP synthase, right? That can allow the protons to come through. So we can generate ATP. So this right here is the agar bridge. It's going to allow ions to move from one half cell to the other. This is the agar bridge, by the way. It would have normally been a salt bridge in Gen Chem 2. The electrons flow from one half cell to the other through the wire that connects the 2 half cells to the voltmeter. And this is so we can actually read the voltage across this, which is the EMF, electromotive force, or another way of saying that, it's the potential energy. Okay. Again, this is going to be a solution. This solution has 1 molarity of X and 1 molarity of X minus. That's that pair where we've gained 1 negative charge on that. This is the standard reference cell, by the way. That is that H plus in equilibrium with H2 gas. This is not open to the air, by the way. It's in a glass ampule or tube, so you don't lose the H2, which is gas. Okay, you guys have actually kind of used this before. You may remember your pH meters. Do you remember your pH meters at all? That's just this. That's just a very specific application of this. The glass being the porous part. So if the reaction proceeds in the direction like so, where we have X minus plus H plus, this gives us the neutral X plus the hydrogen gas. The reactions in the half cell would be these right here, which is the X minus portion gaining an electron, losing an electron, to the X plus the E minus, which is the electron. And the H plus has the gain in electron, which gives it half of the hydrogen gas, which is neutral. Okay. Anybody have any questions about this so far? Kind of coming back? So the electrons are going to flow from the sample half cell to the standard reference half cell, and that's that electron transfer happening. Now remember, in our cases, we don't have a wire. What do we have? We have NADH and FADH2, don't we? Those are our, at least part of our wires, quote unquote. So the sample cell electrode is going to be taken to be negative with respect to the standard cell electrode. Reductant or reducing agent is going to be the donor of your electrons. It's going to be the things that give the electrons, so the other thing can be reduced. Okay? Kind of like if an assassin isn't the one that gets killed, right? Okay. Okay. So a reducing agent isn't the one that gets reduced. It's the one that does the reducing. Still sounds like I'm talking about assassination. Still. Oxidant, or the oxidizing agent, is going to be the acceptor of your electrons. So for example, in the example I gave, it was H+. And that's the one that was reduced. It's going to be the one that oxidizes the other thing, right? Negative E0, E prime 0 means that the oxidized form of a substance has a lower affinity for electron than does the H2. So NADH, for example, is a strong reducing agent and has a negative E prime 0. The positive E prime 0 is going to mean that the oxidized form of a substance has a higher affinity for electrons than does the H2. So for example, a O2 is a strong oxidizing agent. It has a positive E prime. You can think of this as the E prime here. This is a potential, right? And so if you're a negative potential, then you're going to give electrons. If you're a positive potential, you're going to take electrons. And all of these are measured against H2. Okay? The standard H plus going to H2 cell. That is the standard cutoff point. The driving force of oxidative phosphorylation is going to be the E prime 0 of NADH or FADH2, depending on the portion of it, relative to that of O2. And so when we're looking at the oxidative phosphorylation, we're going to be looking at it having the NADH versus oxygen, okay? Or the FADH2, again, versus oxygen, right? So these right here are the standard reduction potentials of some of your different reactions that we've looked at. And again, these N's, does anybody remember your N? Okay. N is going to be the number of electrons that are transferred for each reaction. Okay? So for example, in this reaction here, ferrodoxin, this is going to transfer 1 electron in that oxidation reduction. Okay? That's actually just like our example that we looked at before. If we look up here to the acetate, for example, going to acetaldehyde, this is going to transfer 2 electrons. Notice we have the hydrogen here. We have 2 H pluses going to a whole H2. This transfers 2 electrons for that. We had been looking at half of that reaction before, hadn't we? Only generating half of an H2. Anyways, this is the NAD+. It's actually going to transfer 2 electrons. You guys may not have noticed that, but I had hinted at it numerous times where I kept saying it can be called NADH2+. That's why. Okay? It's actually transferring 2 electrons at that time. The NADP+ also does 2 electrons. FAD going to FADH2, again, is 2 electrons. It's kind of easy to keep track of those because the H2s. Okay? Anyways, these are actually important later on, and we'll cover some of these things later on. This is probably your easiest one to remember. Iron 3+ going to iron 2+ gives you 1 electron transfer. Okay. Okay, so notice these numbers here. Negative. What does that mean? Okay, so 2 H pluses going to H2 here, right there. This is the standard condition, right? And that's why that's 0. So we're referencing the standard half cell versus the standard half cell. Okay? If it has a positive value, and if you ever forget, the easiest way to do on these tables is to look at something you know. So oxygen right here, is that a good or bad oxidizing agent? Good, isn't it? Yeah, it takes off electrons very easily, doesn't it? Its number is positive. And so that means oxygen, positive, we take electrons easily, don't we? Okay. If we have something on the other hand that is negative, we're going to fairly easily lose electrons to that, aren't we? Okay. Okay. So the standard free energy chains, delta G prime, not prime, is going to be related to your change in reduction potential, which is delta E prime 0, by this equation right here. You'll actually recognize this equation from sort of the beginning when we talked about the delta G stuff, but also you may recognize this from your Nernst equations, that sort of thing from Gen Chem. Okay? So this is delta G not prime equals negative N. What did N stand for again? Exactly, the number of electrons transferred. Be careful about that. It's not just moles. Okay? It technically is moles of electrons transferred, but that's because you're assuming you have 1 mole of the substance reacting, right? Yeah. So again, number of electrons transferred. Faraday, F is the Faraday constant. That is this value right here for our standard conditions we would normally see. By the way, that's how you actually get your units fixed up. It's the Faraday constant. If you have access to the Faraday constant with its units, that tells you the formula. See that? So you got to get rid of moles, you got to get rid of volts, right? To get into kilojoules. So you need to get into kilojoules per mole. This right here has to be the number of electrons. This then has to be times Faraday constant. And then times the voltage, voltage being a difference in electromagnetic force. Okay? And so that's actually why that ends up in there. Now, one thing to keep in mind here is that delta G's are negative to be spontaneous, whereas delta E's are what to be spontaneous? Positive, yeah. So your sign has to flip, doesn't it? Yeah. Which is why there's a negative in this formula. Okay, so to calculate the delta G naught prime for the oxidation of O2 with NADH, we'll consider the pertinent half reactions as listed in the table. So right here we have 1/2 O2 going with 2 H pluses, 2 electrons being transferred. That gives us the water. And that's that final oxidation we're going to have in this, right? And that has an E naught 0 right here, plus 0.82 volts. Now, this right here is NAD plus H plus 2 electrons transferred. That gives us NADH here. So if we combine our 2 half reactions, this is going to give us this reaction right here. And so we have oxygen going with NADH. Notice here the H plus right there. And then that's going to give us H2O plus NAD plus. Please note that we had to flip this reaction here to get that, didn't we? See that? So we flipped that second reaction. Okay? And that's what caused the electrons to cancel, and we canceled one of the H pluses off, didn't we? Can everybody see it okay? Okay. Okay, so we got that overall reaction. To get the standard free energy for the reaction, which is negative 220.1 kilojoules per mole, you would take the delta G naught prime. That's going to be equal to the number of electrons transferred, well, negative, number of electrons transferred times the Faraday constant times positive 8.82, which is that right there. This is then going to be plus the negative 2 here again, because we're still negative number of electrons transferred, 2, times the Faraday constant, times, what happened to the sign? Yeah, we flipped the reaction, right? So we're going the opposite direction. That means it has to have opposite sign. One of the things you need to be careful about here is, does anybody know if this is an intensive or extensive property? Anybody remember what an extensive or intensive property means? Okay, so like if I have the weight of an ocean versus the weight of a drop, are they different? Yes. And so that matters to the extent I have, right? If I have a drop, it's going to be different than the ocean. Okay? And that's an extensive property. Delta G naught prime, this is not an extensive property, right? Okay? What about this one right here? E prime. Is that an extensive or intensive property? Yeah, so it doesn't matter. Think of it this way. This is like the height of a waterfall, right? Does the height of a waterfall depend on the amount of water going over it? No. No, so that's going to be an intensive property, okay? It's inherent to the reaction. And so if I have something here where I had to multiply this through, that doesn't actually affect this at all. It's one of those weird things where things change a little bit different than our normal like delta H's and stuff. Okay? Okay. And the easy way to think about this is it's like the height of a waterfall, right? So I could like put more and more water over that, and it wouldn't affect the energy coming off that waterfall, would it? Does that make sense? It won't affect the height of that waterfall. That's what I'm looking at. Or another way of thinking about this actually is associated with the electrons. Okay? Doesn't matter how many electrons you pump over it. That stays the same. Okay. And so, again, that ended up being a positive value right here. We're going to add these together. Note that you don't have to worry about the sign. Do I minus? Do I add? That doesn't matter. Okay? Because you take care of the sign up here by flipping those signs, right? So you can just total them up. Okay, and so that will give us our delta G naught prime. Anybody have any questions of how I did that? Okay, please let me know if I lost anybody on that, because it's a pretty popular problem on things like standardized tests and stuff. So if you get like the MCAT, it's pretty popular. Yeah. It's not as popular as like the amino acids. Definitely memorize those before you do this, but it's popular enough. The energy that's associated with the proton gradient, this can actually be quantified. So delta G would equal RT, the natural log of the concentration 2 over the concentration 1. So the concentration on one side versus the other. Plus Z, Faraday's constant, and then the change in V, which is the voltage potential across the membrane. So again, these are the concentrations of the protons on one side versus the other. Okay. The direction you do this, does it actually matter which direction you do this for? No, because this will tell you which direction it's spontaneous in, right? If you get negative value, it's spontaneous in the direction you did from to, right? If you get a positive value, it's the opposite direction. Okay? Not a problem. So again, Z is the electric charge of the proton. Z actually, interestingly enough, does not actually have to be 1 in all cases, but it's usually 1. Okay? Delta V, again, is the voltage potential across that membrane. It's a very weird way of stating it, but it is. R is the gas law constant. By the way, did you guys notice that this is like straight up Nernst equation here? Just done and rearranged. That's all it is. And T is the temperature in Kelvin. Did you guys recognize this? Yeah, this would have been like delta G naught plus RT, natural log, concentration, reaction quotient usually. Yeah, it's just a rearrangement. So the electrons are going to flow from the NADH to the O2 through the 3 protein complexes that are going to be embedded in the inner mitochondrial membrane. Remember that these complexes will also be parts and joined with our citric acid cycle. Okay? This is not actually a completely separate thing. So the NADH to Q oxyreductase, this is the actual name of the enzyme that is this complex 1. Okay? So that's the enzyme portion that does the work. Well, I shouldn't say does work, but it does the enzymatic, what enzymes do, as it were, the catalysis. So complex 1. This is then going to be the Q cytochrome C oxidoreductase, which is complex 3. And then we have cytochrome C oxidase, which is complex 4. By the way, these names tell you a tremendous amount about this, right? You guys remember the classes of enzymes we talked about before? Was there one that was like an oxidoreductase? Yeah, we're doing oxidations, aren't we? Yeah. And then NADH, you may remember that from the table up here. We had the NADH, right? And then Q. Was there something that maybe would start with Q? Well, because we don't have any other choices. So they're actually different transfers of electrons from groups on here, okay? Those are actually our substrates in each of these cases. And that will greatly help you understand these, is that in each of these cases, we're going to be using that difference in the delta E's to move electrons along and move protons along, okay? And again, the electrons are just going to be used to pull the protons along. We'll have to do a few more things. It'll get a little bit more complicated than that, but that's the overall gist. Now, the electron's going to flow through your complexes 1, 3, and 4. This is going to be highly exergonic, and it's going to power the generation of your proton gradient. By the way, you know how nerves are always being called like, this is electrical, you're moving electricity and all that? This is actual electricity. Okay? Not that bullshit where it's not electricity at all. It's actually electrical potential fields, okay, or polarization fields. This is literally electricity moving through, okay? So flows of electrons. The complexes 1, 3, and 4, these are actually your proton pumps, which are why these ones were brought up first like this, okay? You may have noticed I skipped a number. See that? Yeah, I skipped a number. So succinate Q reductase, this is complex 2 that I skipped. This contains the succinate dehydrogenase from the citric acid cycle. Ooh, they're joined, aren't they? Damn it, Dr. Clausen. Yeah, I'm sorry, I have to subdivide these things. Just imagine if I tried to do this while I was talking about the complex 2 succinate Q reductase at the same time I was talking about the succinate dehydrogenase, right? Yeah, would have been very confusing. Remember that these are, again, complexes. Technically, that complex 2 would incorporate and have the succinate dehydrogenase. That is the citric acid cycle portion of that. And it's going to transfer that electron energy through to the rest of the cycle. Okay? And so this is that wiring that I was talking about. that's going to connect both cycles. It's literally wiring, right? You're transferring electrons through it. The electrons from your FADH2 are going to enter your electron transport chain at the Q cytochrome C oxidoreductase. So the Q cytochrome C oxidoreductase is which complex again? Complex 3. Okay? And so it is easier to remember than you think, right? Complex 2 feeds its electrons into complex 3. Okay? Complex 3 will use that electrons to pump protons. Remember that complex 2 does not pump protons itself. It just feeds electrons from your FADH2 in your citric acid cycle. Now, complexes 1, 3, and 4 actually appear to be associated in a supramolecular complex. Okay. I know. It's okay. So we have the enzyme names, right? And those can denote whole enzymes, but they can also denote domains, can't they? Okay? And so the enzymes are the part of it that are actually going to act. Theoretically, they could actually be cut off on many of these things, but they can be assembled into complexes. We then are going to have the complexes, right? Complex 1, complex 3, complex 4. But these complexes can be assembled into higher order domains, right? And so we're going to have a higher order domain. These are going to be associated together, 1, 3, and 4, and they're going to make a supramolecular complex. Remember that our complexes are going to be named 1, 2, 3, 4 in order so that they arrange in order of electrons hitting them. Okay? So for example, complex 2 needed to be at 2 because it gets electrons and transfers it to 3, right? Complex 1 doesn't really have anything directly, kind of does, but it needs to happen before complex 2. And they're going to overall be in the order that they would be associated in the citric acid cycle going from CoA, acetyl-CoA, down through. Okay? Does that make sense? So remember that overall, they're going to follow the citric acid cycle order. Okay? And that's why they're complex 1, 2, 3, 4, and we are going to go in that. So this is going to, the supramolecular complex is going to facilitate the rapid transfer of substrate from one to the next to the next. It's going to prevent the release of reaction intermediates. Remember, you have a lot of oxidation reductions happening here. That means things are going to be picking up charges, right? And when we talk about these charged intermediates, they're very reactive. So they would easily be able to react to a variety of different things inside of this matrix. So we need to keep all of this very tightly controlled and contained, okay? And so we have a little power facility within a power facility. Or another way of thinking about this is if you have a generator at the power plant, do you just leave all the internals exposed to everything? No, you assemble it up into a nice generator kit and, keep it mostly covered, right? So that you don't screw things up and damage everything. Same thing. All right. So here we have the free energy that's relative to O2. And so what we're going to see is we have NADH. It's going to feed into complex 1. It's going to transfer electrons over to ubiquinone. Ubiquinone, which is Q. By the way, that's the Q. That's the Q that comes up here. The succinate Q, that's the Q that comes here. NADHQ. Notice again, this is actually very standardized formatting. Okay? So we have electron potential going from what? NADH to Q. Okay? And that's why the name, NADH to Q. This is going to be an oxidoreductase. Oxidation reduction, right? Okay. And then we're going to have complex 2. This is going to have the FADH2 coming in, giving the electron density. Now, if you remember, the source of that FADH2, that was succinate, right? And so the electrons are going from succinate, technically through FADH2, to the Q. Okay? Q is going to come in. See, the Q's in front now. Okay? The names tell you almost everything you need. Pay attention to those. Q is going to come in. It's going to transfer electron density to cytochrome C. Again, oxidoreductase, because we have an oxidation reductions happening, right? And then cytochrome C, the next one is cytochrome C oxidase. Now, notice that we don't get an oxygen in here because the name of the enzyme is oxidase. Where do the electrons go? Oxygen. Okay? And so cytochrome C to oxygen. All right. And this entire time, we're going to be going down our potential energy. Our waterfall is pushing. So we get a nice collection of electrons and protons on the other side, right? So we're pulling those protons through to the other side. At this point, we would have built up proton density. All right, does anybody have any questions about that overall concept? Okay, that'll give you the lens to look through this. So these right here are the components of your mitochondrial electron transport chain. Again, here's the enzyme complex, NADH to Q, succinate to Q, Q cytochrome C, and cytochrome C. This right here is your masses, by the way. These are the prosthetic groups. Remember the different prosthetic groups we had talked about? This is the FMN right here. This is the Fe to sulfur. Those are usually assembled into complexes, though, which we'll see, where it's more than just one Fe and S. This is the FAD that we had just mentioned. This is heme, but they're specialized hemes. They're not the same. They're not exactly the same as you would see in hemoglobin. Okay? So this is going to be heme, BH, BL, and C1. This right here is your heme A3. This actually has copper A and B. Okay? And so one of the things that's going to help transfer these electrons is which things can lose and gain electrons fairly easily, and we'll use in wires because of that. Metals. Specifically iron and copper wiring, right? Guess what? Iron and copper wiring. It's just that in this case we have to modulate their ability to lose and gain electrons. And so we're going to have things that are bonded to them, like sulfur, or we're going to have these heme groups. This right here is the matrix side of this. This would be NADH on the matrix side, and this would be succinate on the matrix side. This is the membrane core. What do you think the membrane core refers to? The thing that's going to move around on the membrane, right? Okay. This is Q. Anybody remember the name of Q? Ubiquinone. Okay. You can't use the UB part. It wouldn't make any sense because there's other U stuff involved. So you get the quinone. Q. Okay. And then we're going to have here the intermembrane side. Intermembrane side being the what side? Side where we're building up the protons, right? Okay. And so this is going to have the cytochrome C and cytochrome C again. Again, those are both the ones that say cytochrome C. Okay? As long as you remember those names of the enzymes, everything else falls really easily. So the coenzyme Q, or standing for ubiquinone, or coenzyme ubiquinone, this is the hydrophobic quinone that's going to diffuse rapidly within the inner mitochondrial membrane. It's derived from isoprene. This can exist in several different oxidation states. This will couple the electron transfer reactions to the proton binding and release. Your coenzyme Q is going to function as your electron carrier. So it's going to reduce from the carries electrons from complex 1 to complex 3. So again, you reduce it, it carries the electrons. Reduced means extra electrons on, right? The transfers electrons from the FADH2 from the citric acid cycle to complex 3 as well. And that would be the complex 2 portion of that. I think I'm out of time, right? Okay. You all have a wonderful day. We'll start up on this. Hopefully you'll remember the quinone portion of this that we just went through. Because that'll be your different oxidation reduction states. Okay, I will see you all on Wednesday. If you haven't watched that lecture, please watch the Sunday lecture today or tomorrow. And don't forget, I had to get rid of some of the chapters. They become extra credit. And so check that out.